I'm Daniel Levine, and this is the Bio Report. The enzyme GSK3 beta in healthy cells is involved in glucose metabolism. In cancer cells, though, it serves as a master regulator of tumor growth, progression, and cell survival. While GSK3 beta has long been an attractive target in cancer therapy, it's been difficult to inhibit because of the poor pharmaceutical characteristics and adverse effects of therapeutic candidates. Actuate Therapeutics' experimental therapy, elraglusib, has shown early promise. Results suggest it not only suppresses tumor growth, but also activates the immune system to combat cancer. We spoke to Dan Schmitt, President and CEO of Actuate, about elraglusib's potential to overcome chemoresistance, its recent clinical success in metastatic pancreatic cancer, and the drug's unique multimodal mechanism. Dan, thanks for joining us.
Oh, thank you. I appreciate the time and attention.
We're going to talk about cancer, the kinase GSK3 beta, and Actuate Therapeutics' efforts to develop a therapy that can inhibit this. Let's start with GSK3 beta, though. This is a kinase that can inhibit the growth of certain tumors, but in other cancers, its overexpression can drive the spread and growth of cancers. What is GSK3 beta's role in healthy biology, and what is its role in various cancers?
So there's appreciate the question, and there's been an evolution in the understanding of GSK3 beta, both in normal cells and in cancerous cells. And the dogma has shifted as science has progressed. So in normal cells, GSK3 beta is expressed at extremely low levels. It's constituently activated, and it's resident in the cytoplasmic domain of cells. And there it's largely involved in metabolic processes, particularly in glucose metabolism, thus the name GSK. In cancer cells, it's been shown to have been co-opted and translocated from cytoplasmic domain into the nuclear compartment. And there it sits upstream of some, it's sort of a master regulator across some key oncogenic processes. Now, in the, 10 years ago, there was a whole set of data or hypothesis that this was actually a tumor regulator, and if you inhibited it, you would see tumor growth and progression, but along the Wnt beta-catenin pathway. But that's largely been addressed and dispelled. GSK3 beta in cancer cells, it sits upstream of NF-kappa B. Now, NF-kappa B is notorious in cancer. It's responsible for driving gene expression that regulates things like tumor growth and progression, chemoprevention, and anti-apoptotic processes, anti-cell death. So you're basically making cells immortal. What's been shown over the years is that, it's very difficult to target NF-kappa B directly. The key to that lock fits a lot of other locks. And so there's always a lot of off-target effects by trying to target NF-kappa B. But what's been shown is that downregulating or inhibiting GSK3 beta results in a downregulation or an inhibition of those NF-kappa B mediated processes. So that's really where we started our program, is that relationship and looking at refractory cancers, which means they don't respond to chemo treatment, and the role of NF-kappa B in that process, and then selecting those cancers where that's the major driver of chemoresistance, and then targeting it with GSK inhibition. And we've shown through those early years of research that it is in fact doable to reverse that chemoresistance in those refractory cancers by inhibiting GSK3 beta.
You mentioned this has been difficult to target because you often end up hitting other targets. How have you gotten around this? How have you been able to achieve specificity?
Well, so NF-kappa B is notoriously difficult to target. GSK3 beta, the medicinal chemist set out to design highly specific GSK3 inhibitors that were not only specific but potent, as well as highly lipophilic, because the medicinal chemist was focused on this target in CNS disease. So there's been a lot of literature and literature recently suggesting that GSK3 is an active moiety in Alzheimer's disease as well as bipolar disorder. And lithium is actually a GSK3 inhibitor. Hits a lot of other targets, and it's a very dirty drug. So the inventor was trying to make a much more specific inhibitor and potent and lipophilic, so it would cross the blood-brain barrier for CNS disease. Just so happens those same attributes play well in oncology. So it's interesting that when we licensed the portfolio, there were over 100 different compounds on the same pharmacophore, and Elra was highly specific. The first two in the kinase screen, the first two that is most potent against are GSK3 beta, alpha and beta, and then the next four just happen to be all oncogenic kinases. So FLT3, PIM, MINC, and MAP kinase. And then everything else on the kinase screen is at least a log or two higher in its IC50s. So we have a small molecule, highly specific agent that's very potent against the target, but the next set are all oncogenic as well. So that may play into how well this drug behaves.
Given the role that GSK3 beta can play as a tumor suppressor, is there any concern that targeting and inhibiting it for one cancer could actually lead to other cancers?
We have not seen that. And I think that GSK as a tumor suppressor has lost its scientific founding, right? I mean, there was a very 10 or 15 years ago, that was the thought, as GSK sits in this Wnt pathway. And we have seen no actual data suggesting that is still the case. As the science has progressed and as more research has been placed on GSK, I think that sort of tumor suppressor role, it really has faded into the mist.
Well, Actuate's lead experimental therapy is a GSK3 inhibitor. What is it and how does it work?
So when it's interesting. So it's a very unique multimodal mechanisms of action agent. So as I said, when we first started into the clinic, we were focused on those cancers where NF kappa B was mediating resistance, and so we were dosing um, patients who were shown to be refractory to chemotherapeutics, where that mechanism of chemoresistance was known to be mediated by NF kappa B. And what we would do is we would enroll those patients into our phase 1b/2a trial. We had a very large 8-arm basket trial. And all the patients enrolled were shown to be refractory to chemo and then re-challenged with that very same standard of care plus elra. And the idea was to show one of two things. One is, are there any new safety signals associated with putting elra on top of those standard of care chemotherapeutics? And two, was there any evidence of increased efficacy in patients who were previously refractory when they were re-challenged with standard care plus elra? And so, and the evidence came out that yes, indeed, no new safety signals, but all evidence across all 8 arms of that basket trial, all 8 different standard of care chemotherapeutics showed evidence of increased efficacy in previously refractory patients. And that's really where we started our clinical development path. Now, a lot's been learned in the clinic since then. Um, in addition to the NF kappa B mediated processes, there's a significant amount of credentialing research that shows that in pancreatic cancer, for example, downregulation of GSK effects is active on the DNA damage repair pathway, as well as epithelial to mesenchymal transition. So mesenchymal cells in pancreatic cancer are highly resistant to treatment. Inhibiting GSK has been shown to transition that mesenchymal phenotype back to epithelial, and then the cells become susceptible to chemotherapy. So there's a multitude of mechanisms on the reversal of chemorefractory cells to a chemosensitive type. What we've seen in the clinic since then, since being in the clinic, is that downregulation of GSK also upregulates immune response. And it's very interesting. There was a paper that came out last fall in Nature Frontiers, a Nature publication, where the investigators showed that either through GSK knockout or through use of elra, tumor cells were upregulated for neoantigen presentation on the surface of the tumor cells, basically making themselves visible to the immune system. We call it turning cold tumors hot. In our clinical trial at Brown, investigators had a cohort of refractory metastatic colorectal patients, and in those patients within 1 or 2 doses of elra, there was a significant increase in the activation and mobilization of immune cells. So an expansion in the numbers of both NK and T cells in these patients. And in that T cell population, there was a significant portion focused on a single receptor. And we believe, based on what we're seeing in the clinic, that receptor's associated with the tumor itself. Just recently at ASCO, we had a major presentation on oral presentation in a plenary session on Saturday, which is sort of that's the prime time slot. That in tumor biopsies in patients who received the standard of care Gemabraxane plus Elra, there was a significant numbers of both CD8 positive and granzyme B positive T cells in their tumors themselves, a significant increase pre-treatment versus post-treatment. And that increase was not evidenced in the control patients that got Gemabraxane alone. So you got this stream of upregulation of neoantigen presentation, expansion of immune cells in the bloodstream, and then actually a recruitment of those IO cells, those immune cells, into the tumors of patients themselves. So it's a complete sort of picture that this agent not only affects, downregulates tumorigenic processes, but it also upregulates immune processes. And that's why we think we're seeing the kind of survival benefit in our patients that's, statistically significant responses in a very difficult to treat cancer.
This is being developed both as an injectable and in oral therapy.
Yes.
Why the two formulations?
Well, I think that there are some patient populations where an IV makes a lot of sense. So current standard of care in pancreatic cancer is intravenous. Fulfirinox, Gemabraxane, they're all IV. Patients come to the clinic. It also was the fastest way to the clinic to manufacture and test this agent. With that success, that proof of concept that this is active and it's a very benign drug, the decision was then to make investments into developing an oral formulation. And what the oral formulation does is it allows us to dose into patient populations that typically don't come into the clinic, and also expand the dosing regimens from a weekly IV infusion to perhaps a daily dose for the treatment and management of cancers. So it's going to be a product line.
You mentioned that this can help enlist the immune system in
Yeah.
fighting cancer, turn cold tumors hot. Are you thinking about this as a monotherapy, or does that suggest certain combinations?
Well, I think the answer is yes and yes. So we have some pretty profound responders on monotherapy, particularly those with refractory metastatic melanoma. We have, it's a terrible, terrible disease. Once it goes metastatic, it tends to run throughout the body. And we did a small study as a phase 1 study leading to the basket trial. Just so happens we had patients with refractory metastatic melanoma tested in the monotherapy arm who had complete responses and are still living 6 years later from a prognosis of months, weeks to months. So there may be an opportunity to develop it into those cancers that we know are exquisitely sensitive to the downregulation of GSK3. We also know there are a number of cancers where the combination makes a whole lot of sense. And we believe, based on mechanistic profiles, that this drug can be combined with PD-1 inhibitors and RAS inhibitors, potentially MEK inhibitors, that in addition to the standard of care chemos, where we think we can enhance the efficacy of both by shutting down compensatory pathways. So I think that a lot of cancers are going to continue to be combination treatment, and we will and give the patients the best chance for sustained and quality of life. And I think that it's contextually different across different histologies of which ones will go after monotherapy and which ones will stay combination.
This is a target that's implicated in a broad number of cancers. Your lead indication is pancreatic cancer. Why start there?
I get that question a lot. Um, it's a very difficult to treat cancer. So when we started, we had a lot of preclinical information, like every program does, um, that pointed to specific combinations where there were synergies. And gemcitabine is one of those where there was profound synergy demonstrated preclinically. In the 8-arm basket trial, we saw synergy there in refractory patients as well, across and other histologies as well, such as non-small cell lung cancer, metastatic colorectal cancer, glioblastomas. Um, but the the culmination of that basket trial and the ability to leapfrog the development program into a first line setting was really attractive, uh, not only to us, but to patients as well. And we've demonstrated significant clinical benefit in these first line patients. So it's really that ability to move this to a first line setting that sort of made it outweigh the other histologies that we have on our target list.
Well, what's known about it from the studies you've conducted to date in pancreatic cancer?
Well, we've done two separate studies in first line patients. The first study was a single arm proof of concept trial. We recruited patients. We recruited approximately 40 patients, but the design was we would recruit up until the 23rd patient got their second scan, and that means they became evaluable. And then we stopped recruiting and we treated and tracked this population. And it was very interesting because it's assignment 2 stage. You set a futility goal in the middle of the study. And this futility goal was to have a disease control rate, which means, what percent of your patients, their tumors either stayed stable or reduced. And the historical control was a 50%. And if we exceeded 50% disease control, then we would open up the second half of the study. But the data from the first half was so compelling, in terms of, we had a median overall survival of over 15 months, um, in our evaluable patients, and the historical control had 8.5. And even if you take all the patients that were enrolled, whether they got treated or not, or were evaluable or not, we still had a median of 11.9 months versus 8.5. We had 3 CRs in 23 patients. The historical control had 1 in 431. So it was like, okay, very compelling data. The agent actually is showing that it is working in a very meaningful way on meaningful clinical endpoints. So instead of opening up a second half of a single arm study, we felt we met the proof of concept. We next initiated a very large international randomized phase 2 study that compared, in first line treatment of metastatic pancreatic cancer, patients treated with gemabraxane as our control versus patients treated with gemabraxane plus elra as the combination group. And the day we ran this study, it was 286 patients recruited from 60 clinical trial sites across the world. And the results were presented at ASCO this year, showing a statistically significant increase in median overall survival, almost a 3-month increase in median overall survival, 10.1 versus 7.2 months. We also doubled 1-year overall survival in this patient population, 44 versus 22. And that survival benefit continues down from 12 months to 18 months to 24 months. And that's in the total study. At the time we presented the data, only 10% of the patients in the control arm, the standard care arm, were alive versus 26% in the combination arm. And we looked at subphenotypes, sort of looking for if there were any subgroups that did not respond. And all of the analysis point to statistically significant increases in survival in the combination group. The interesting thing is we did this with a very benign side effect profile. So the arms were balanced coming into this trial for demographics and disease status. Going out of the study, they were balanced in terms of the percent of patients that go into second-line therapy and the type of second-line therapy. And the only difference between the two arms was that one got elra and did significantly better. From a safety perspective, there were only 2 safety signals that were, noted as higher in the combination group than the control. One is an ocular event. It's very transient. It was seen with previous GSK inhibitors as well. And basically, patients, while they're getting an infusion and up to a day later, report their visual field turns a little bit on the blue to gray side, almost like a color blindness. And what it is GSK is resonant in the photoreceptors and the rosin cones. And when you inhibit it, you lose sensitivity to light, particularly in the lower part of the spectrum, the red part. But within 18 to 24 hours, it completely comes back to normal. There's no intervention. Physicians noted as a, sort of a nuisance side effect. There's no quality of life issues. And it's completely transient. The only other safety signal higher in the combination group was grade 3/4 neutropenia. This is a well-known phenomenon associated with Chemoresistance, and physicians are very, used to managing it either through use of Neupogen or down, dose reduction on Chemoresistance. Interesting thing is, while we had an increase in neutropenia, we did not have increases in either febrile neutropenia or sepsis. So we had an increase in the incidence of a decreased lab value. Patients don't feel it. It doesn't affect their quality of life. So we really have a very unique agent that either, it potentiates the ability, survival 2 to 3-fold a year and beyond with exceptionally minimal side effects. And it's just being added right on top of standard of care. So if a patient is thought to be a candidate for Chemoresistance, this is just added on top as sort of a backbone.
What's the development path forward?
Well, we're working on several programs. We see that metastatic first-line treatment of metastatic PDAC is definitely one of those that's on the registration path. And we'll be working with the FDA to get clarity on what's required beyond this study to register it for that indication. We also have a program in Ewing's that we had a small pediatric study of all comers to test the drug out in some very areas of high unmet need, neuroblastoma and sarcomas in pediatrics. And it just so happens we had a number of patients with refractory Ewing's that like the first 2 patients had CRs with a complete response. In refractory, there are 2 standard lines of care for Ewing's sarcoma. And if you're refractory to those, there's really nothing. The disease becomes 100% fatal. And so the pediatric oncology community is exceptionally interested in this drug because they don't see CRs in that patient population. And we had 2, the first 2 patients coming in had complete responses. So that's on the development path. When the oral goes into the clinic, we'll be looking at treating metastatic melanoma as a monotherapy, potentially. And then either one or both of metastatic colorectal cancer and or non-small cell lung cancer, where we have clinical data that shows significant benefit in early studies.
How broad a set of cancers might this be useful in treating, and how are you prioritizing the indications you'll pursue?
Well, it's a matter of those where we think we can have the most clinical benefit, right? Based not only on mechanistic data, but um, clinical data that's informing us now. You also look at, you look at the competitive space and what patients are available to be enrolled into clinical trials. That weighs on it as well. So there's a set of factors that dictate which which ones will be first and which ones will follow on. And typically it's where do we think we can have the greatest impact and the greatest number of patients.
Actuate raised 17 and a quarter million in a public offering in September. How far will existing cash take you?
We project that'll take us through at least the middle of next year.
And how are you balancing clinical development with cash in the current financing environment?
I think that's the challenge of every biotech company and biotech CEO, right? So we have laid out our development path with key catalysts. We're watching the financial markets for the response to different factors, and we are always opportunistic when looking at raising funding. We do it in a way that's investor friendly, but we keep our eye on uh, developing the programs because investments required. Um, you don't want to be too tied to avoiding dilution. You really want to be tied to making progress, and that's where you really get um, benefit for everybody that's involved, investors and patients and treaters as well.
Well, what's the discussion with investors like these days, and are you noticing any change towards the industry as a whole?
Oh, I think so. I think so. Remember, not too long ago, I think the XBI was in the 80s, right? The biotech index, and I think it's well over 100 as we speak. Um, so there has been a refresh of interest, and I would say it's just been since September that there's been a warming of the markets to the biotech sector. See a number of deals being done. You see a number of financings being done. It's been, it's sort of the market goes in cycles, and I think we're finally getting some, positive momentum in terms of the investment community in these types of programs.
Right now, I'd call Actuate a pipeline and a product company.
Right.
Are there plans to bring in other candidates into clinical development, or are you working on expanding trials for ERLA?
Well, we're opportunistic, right? So if the right candidate came along, we felt it was synergistic with our capabilities and our pipeline, and we could fund it, appropriately. We certainly would take a long look at that. I mean, we have a lot of expertise across the board in our organization. Our primary focus is to get this product on the registration path and get it to successful execution either through us or through collaborations, strategic collaborations.
Uh, are you looking for a partner to commercialize this, or would you expect to do this on your own?
I think we are fully capable of doing it on our own, but we would be, we're always in discussions with potential pharma because really this has the potential to be a franchise either for us or for a big pharma. So I certainly wouldn't want to, paint myself into to a corner and say we're only going to do it with pharma because that's not a smart strategy for a company. So we're building as we go, and if somebody comes along with the right terms, we're open to discussion.
Dan Schmitt, President and CEO of Actuate Therapeutics. Dan, thanks so much for your time today.
No, I thank you. I appreciate the interest and your time as well.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.